Hello everyone, I am Prajwal Ghatal and today we will discuss a very interesting and rare topic called Wilson's disease. Wilson's disease is an autosomal recessive disorder of copper metabolism. The disorder leads to excessive absorption of copper and deposition of the dietary copper into the liver, kidney, brain and other tissues. The presentation can widely vary from chronic hepatitis to asymptomatic cirrhosis, acute liver failure to neuropsychiatric symptoms with cognitive impairment. When we talk about the signs and symptoms of Wilson's disease, the signs and symptoms of liver include swelling of the liver, jaundice, yellowing of skin and the white of the eye area, fluid buildup in the legs and abdomen, a tendency to bruise easily, and fatigue. The neurological symptoms include problems with speech, swallowing and physical coordination, tremors, uncontrollable movements, muscle stiffness and behavioral changes. The other signs and symptoms of Wilson's disease include anemia, low platelet or white blood cells count, slower blood clotting measured by blood test, high levels of amino acid, protein, uric acid and carbohydrates in urine, and premature osteoporosis and arthritis. Another very specific clinical symptom of Wilson disease is Kayser-Fleischer rings, also known as KF rings. These rings are golden brown rings, generally lighter than rust, that forms around the edge of the iris and cornea. They appear in both the eyes and are generally caused by copper deposits in stroma. When we talk about the age specificity of the symptoms generally appear between the ages 6 to 40 and can start early as 2 and as late as 72. The symptoms can also vary and depend on the diet you eat. You may be wondering that these are the signs and symptoms, but what is Wilson's disease? How it is caused? So now let's discuss it. Wilson's disease is caused by the mutation in the Wilson's disease protein. This protein is present on chromosome 13 and it also has a name of ATP7B copper binding protein. It has various functions and one of its specific function is the removal of excess copper from the body. The inheritance of Wilson's disease is autosomal recessive. We take approximately 1 to 2 milligrams of copper per day. That is for our daily needs. But actually our body needs only 0.75 mg of copper. So the excess copper that is in the body is excreted out from the body. It is done 90% in the bile and 10% by urine. The bile excess copper is excreted as fecal copper and the other 10% is excreted as urine. In Wilson's disease, there is because of genetic defect, the excretion of copper is not proper and there is excess copper inside the body, and this excess copper accumulates in various tissues. Now in the tissues, just as iron, copper also has a tendency to react with hydrogen peroxide to form free radicals, and these free radicals are also harmful for the tissue and cause further tissue damage. Okay, so now when we consume copper, it is absorbed from the intestines or from the enterocytes, and from the enterocytes through the portal vein it reaches to the liver. So liver is a very important organ here. Liver stores copper, and in liver copper is sent to a specific protein called A7B, that we discussed earlier. This A7B is very important as it is responsible for two functions. One of the function of ATP7B is that it binds copper to the apocellular plasmin protein. This apocellular plasmin protein is responsible for 95% of copper transportation in the blood. After binding of copper to the apocellular plasmin, it is called as cellular plasmin. 6 molecules of copper can bind to the 1 molecule of cellular plasmin. Then another function of ATP7B is that it is responsible for packing the excess copper into the vesicles, and these vesicles are excreted into the bile, and from bile it is excreted out of the body. In Wilson's disease, you can guess that due to the autosomal recessive defect in ATP7B transport protein, the the binding of copper to cellular plasmin is not done, and also the excretion of copper through vesicles is also not done. As either of the function is not happening, this leads to the buildup of copper in the hepatocytes and also leads to the increased free radical formation. Because of the damage of the hepatocyte, from there the copper and the excess copper and the free radical goes into the interstitial space, and from the interstitial space it enters to the blood vessel. In the blood vessel there are RBCs, so this free radical directly act on the erythrocytes and cause its damage, and from the blood it also goes to various other tissues, causing damage to the respective tissues. Along with these tissues, there is one particular organ that is affected, and that is brain. There is damage in the brain can cause many neurological symptoms and complications. Depending on the location of the damage, the various disorders can be seen. If it deposits into the basal ganglia, it can cause movement disorder, which are similar to Parkinsonism, and if it gets deposited into the cerebral cortex, then it can be toxic to neuron and which can lead to neuronal cell death and dementia. If it gets deposited into the eye, in the decimates membrane of the cornea, then which is between the endothelial layer and the stroma, then we can see KF rings in the eyes, in which there is copper accumulation in the cornea of the eye. As the liver damage occurs the earliest, then we can therefore we can see acute hepatitis and later cirrhosis in liver. In the blood, we can see less concentration of the cellular plasma, increased concentration of the copper in the blood, and also increased concentration of copper in the urine. Along with this, we can also see the enlargement of spleen and liver, and also hemolytic anemia due to the direct attack of free radicals on the RBC, and thus destroying the RBCs. So this was the lecture for today. The treatment options we will discuss in another video, and hope you liked the video and you understood it. Thank you.